Hi, hello, welcome to Neat with Kiran sir. So, students, today we are going to discuss another important chapter of class 12, that is sexual reproduction in flowering plants. So, remember, students, when I'm going to talk with this chapter, you have to have some basic concept regarding the what are the morphological character of a flower. Okay, so for that, you can go through a chapter called as morphology of angiosperm. In that we have to start with inflorescence, flower, the different parts of the flower, different modification. Okay, these things are very important in this beginning of this chapter. Similarly, when I talk with the plant kingdom, we have already studied regarding what the important general characters of the angiosperms. At the last of that general character, we also discuss. So, what do you mean by pollen grain? What do you mean by pollination? Okay, what do you mean by embryo sac? How many cells are there? All the things and all these aspects we are going to study about an important chapter called as sexual reproduction in flowering plant. Okay, my dear students, so here we'll begin with the introduction. So, what is flower is? So, when I say flower nothing but it's a modified shoot. Flower is a modified shoot which is meant for a sexual reproduction. So, remember the flower is one of the important character of the angiosperm where it is going to differentiate between the gymnosperm and angiosperm. So here angiosperms are generally known as the flowering plants because these are the only group of plants they can produce a flower and specially meant for the sexual reproduction. So then we'll go with in depth here. So when I say how a shoot is modified into a flower, so remember when the plant attains the reproductive maturity, or you can say when it completes the juvenile or vegetative phase, it enters into a reproductive phase. At this time, ಇಲ್ಲೇನಾಗ್ತದಪ್ಪ ಅಂತಂದ್ರೆ there is a change in the hormone. So there is an hormone called as florigen. Okay, there is an hormone called as a florigen which converts the shoot apical meristem into floral meristem. Shoot apical meristem into a floral meristem where a shoot is get modified into a flower. Then in brief, when you're going to deal with a flower, so remember flower having a four different orals, that is outermost we call it as calyx, the unit of calyx is called as sepals. Next to or inner to the calyx we have the corolla, the unit of corolla called as the petals, which are brightly colored oral of the flower which are meant for the attraction for the insect for the purpose of the pollination. Then these two, that is calyx and corolla together, we are going to call it as what, accessory orals or non-essential orals because they don't take part in a sexual reproduction and they are meant for the production purpose. Okay, similarly when I come for the third oral, that is called as the androecium, and the innermost oral we call it as a gynoecium. So when I say androecium comprising of the group of stamens and each stamen having an anther and a filament. So androecium represents the male part. So here in the stamens we find anther and filament, anther having a fertile part which contain a microsporangia where the pollen grains are produced, and filaments are referred as a non-fertile part, they don't take part in a production of the pollen grain. Similarly, when I come for gynoecium, that represents the female part. So here in that female part we find a group of carpels or pistil, and each carpel or pistil having three parts, that is stigma, style and ovary. So inside the ovary we find the ovals, and inside the ovals we find the embryo sac which act as a female gametophyte, you can say a female gamete. Okay, is it about the brief introduction regarding a flower? So then we'll go for the structure and the development of anther. So remember when I say when the anther is going to develop, so here we are going to observe at young anther, the anther is covered by the four important wall layers. So before that, so when I talk about anther, typically the anther is made up of four lobes. Okay, how many lobes here? Sorry, two lobes, that is called as dithecous anther. So the anther is said to be dithecous anther where the number of lobes are two, and each lobe we find two sporangia. Okay, each lobe we have two sporangia, hence in a two-lobed anther we are having to observe the four microsporangia. Since it is about all this structure, hence we are going to call it as what, dilobed, or you can say bilobed, or you can say dithecous. Okay, dithecous, how many sporangia here? Four, hence it is called as tetrasporangiate. okay, dithecous and tetrasporangiate anther. So if it is monothecous, sometimes you can expect a question based on the number of lobes. Okay, if an anther having only one lobe, then it is said to be monothecous and bisporangiate, because in a monothecous anther we are having only two sporangia. Okay, this is about the brief. Then come to structure of an anther. So anther is surrounded with a four wall layer. Generally they are called as anther wall. So the outermost anther wall we call it as epidermis, which is made up of a single layered parenchyma cell meant for production. Then inner to the epidermis we are having an endothecium. So that endothecium is a radially elongated cell having a fibrous thickening. Okay, radially elongated cell having a fibrous thickening, and they are hygroscopic in nature. They helps in dehiscence of anther when the anther become matured. Then inner third layer, that is we are going to call it as middle layers, or generally also known as middle wall layers. So these middle wall layers, okay, so it is having 2 to 3 layer, you can say 1 to 3 layers of a compactly arranged cell. So these are going to appear, or you can observe or visualize these layer when the anther is young. When the anther become completely mature, these layer to be crushed, and okay, they are appeared in the form of a crust format or crust manner, crush agirtave. Okay, the innermost layer, that is we are going to call it as tapetum. Tapetum meant for the nourishment for the developing pollen grain inside the pollen sac. So here sometime you can get a question based on arrange the anther wall layers from inner to out or outside to in. So remember, when I go for outside to in, so here the outer one is epidermis, endothecium, middle layer, and innermost is tapetum. Or from inner to out, tapetum, middle layer, then endothecium, and last one is epidermis. Then how can you define a microsporangia here? Microsporangia, nothing but what here? It is a core part which is encircled by a tapetum, or you can say a region which is surrounded by a tapetum. We are going to call it as microsporangia. It is having a sporogenous tissue, and these sporogenous tissue consisting of the pollen mother cell, and these pollen mother cell undergo meiotic cell division. They get converted into haploid pollen grains. Okay, then so here in the anther part only we find at the center, that is at the center of the forelobe, we find a structure called as connective. That is nothing but simply the extension of the filament. Okay, that is the extension of a filament. We are going to call it as what? The connective, which consisting of xylem and phloem. Okay, which consisting of xylem and phloem. Okay, so that will meant for the supply of food and the water for developing anther. Once again ಹೇಳ್ತೀನಿ ಕೇಳ್ಕೊಡ್ರಿ. ಸೊ ಆಂಥರ್ ವಾಲ್ದಲ್ಲಿ 4 ಲೇಯರ್ ಇರ್ತವೆ. ಔಟರ್ ಮೋಸ್ಟ್ ಎಪಿಡರ್ಮಿಸ್, ಅದರ ನೆಕ್ಸ್ಟ್ ಯಾವ್ದು ಬರ್ತದೆ? ಎಂಡೋಥೀಶಿಯಂ. ಎಂಡೋಥೀಶಿಯಂ ಕೆಲಸ ಏನಪ್ಪ ಅಂದರೆ ಆಂಥರ್ ಡಿಹೈಸ್ ಆಗಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ, ಒಡೆದು ಪೋಲನ್ ಗ್ರೇನನ್ನು ರಿಲೀಸ್ ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ಅದಾದಮೇಲೆ ಮಿಡಲ್ ಲೇಯರ್, ಅದ್ರ ಫಂಕ್ಷನ್ ಗೊತ್ತಿಲ್ಲ, ಅದು ಹೆಂಗೆ ಆಂಥರ್ದಲ್ಲಿ ಮಾತ್ರ ಇರ್ತದೆ, 1 ರಿಂದ 3 ಲೇಯರ್ ಇರ್ತದೆ. ಇನ್ನರ್ ಮೋಸ್ಟ್ ಲೇಯರ್ ಯಾವುದಪ್ಪ ಅಂದರೆ ಟಪೆಟಮ್, ಅದ್ರ ಕೆಲಸ ಏನಪ್ಪ ಅಂತಂದರೆ ಪೋಲನ್ ಗ್ರೇನ್ಗೆ ನರಿಶ್ ಮಾಡ್ತಾ ಹೋಗ್ತದೆ. ಇದು ಆಂಥರ್ ವಾಲ್ ಲೇಯರ್ದು ಬೇರೆ ಬೇರೆ ಕೆಲಸ ಆಯ್ತು. ಸೆಂಟ್ರಲ್ ಕೋರಿಗೆ ಬಂದರೆ ಯಾವ ರೀಜನ್ನು ಟಪೆಟಮಿಂದ ಸರೌಂಡ್ ಆಗಿರ್ತದೆ, ಅದನ್ನು ಮೈಕ್ರೋಸ್ಪೋರೋ ಏನಂತಾರೆ, ಸ್ಪೊರಾಂಜಿ ಅಂತ ಕರಿತೇವೆ, ಮೈಕ್ರೋಸ್ಪೋರ್ ಅಥವಾ ಏನಂತ ಸ್ಪೋರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ಸೊ ದ್ಯಾಟ್ ಹ್ಯಾವಿಂಗ್ ವಾಟ್ ಸ್ಪೋರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಅಂತ ಇರ್ತದೆ. ಅದರಲ್ಲಿ ಪೋಲನ್ ಮದರ್ ಸೆಲ್ ಇರ್ತವೆ, ಅವು ಮುಂದೆ ಏನಾಗ್ತವೆ? ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ತವೆ. ಸೊ ಇದು ಒಂದು ಫಂಕ್ಷನ್ ಗೊತ್ತಾಯ್ತು ಇದ್ರ ಬಗ್ಗೆ, ಟಪೆಟಮ್. ಸೊ ಟಪೆಟಮ್ ಅಲ್ಲಿ ಇನ್ನೊಂದು ಫಂಕ್ಷನ್ ಅದೇ. ಸೊ ಏನಪ್ಪ ಅಂತಂದರೆ ಇಟ್ ಆಲ್ಸೋ ಹೆಲ್ಪ್ಸ್ ಇನ್ the secretion of what? The sporopollenin. So that is an organic material which is present on the outer surface of the pollen grain, or you can say the exam of the pollen grain. So that is called as the sporopollenin. It is also secreted by the tapetum. So hence we call it as the what? The tapetum has this two function. One is in the nourishment, second one it also act as a secretory in function. Okay, this is about the anther. Then when I come for the structure of pollen grain, so remember the first point, the pollen grains are measured up to 25 to 15 micrometer in diameter. They are spherical in shape. They are surrounded by two wall layers. Outer is exine, inner is intine. Exine is made up of what? The sporopollenin, that is the most resistant organic material which cannot be degraded by any enzyme present on the earth. And here still that the enzyme is not identified yet, the which going to digest the sporopollenin present on the pollen grain. So since of these sporopollenin, we are going to observe different array. So some we are going to observe spiny structures on the pollen grain, some they are having smooth surface, means the different arrays of structure we are going to observe because of a sporopollenin. Then on the exam where there is no sporopollenin, so we are going to observe what? The germ pore. So that is a germ pore through which a pollen tube emerges out from the pollen grain. Then inner layer we call it as intine. Inner layer anta karitare, intine anta karitare, which is made up of pectocellulose, or you can say cellulose and pectin. So it is itself a cell wall of what? The vegetative cell. So means the inner content of the pollen grain we have two cells. One is the larger cell called as vegetative cell, and again smaller we are going to call it as generative cell. So when I say vegetative cell, the cell wall of vegetative cell itself nothing but the entire wall of a pollen grain. Mark it is very important here. The cell wall of pollen grain itself is the cell wall of the vegetative cell itself. It is the outer innermost layer, that is an intine layer anta karitare. So through which the pollen tube will be produced. Okay, the smaller cell we can say it is amoeboid in shape, which is floating inside the vegetative cell or a bigger cell, that is called as generative cell, which undergo mitotic cell division to form what? The two male gametes. So that is a brief structure of pollen grain. ನೆನ್ಪಿಟ್ಕೊಡ್ರಿ, ಸ್ಪೆರಿಕಲ್ ಇರ್ತದೆ, 25 to 50 ಮೈಕ್ರೋಮೀಟರ್ ಇನ್ ಡಯಾಮೀಟರ್ ಇರ್ತದೆ. ಎರಡು ಲೇಯರ್ ಔಟರ್ ಎಕ್ಸೈನು, ಅದು ಸ್ಪೋರೋಪೊಲಿನ್ ಆಗಿರ್ತದೆ. ಎಲ್ಲಿ ಎಕ್ಸೈನ್ ಅಬ್ಸೆಂಟ್ ಇರ್ತದೆ ಅದಕ್ಕೆ ಜರ್ಮ್ ಪೋರ್ ಅಂತ ಕರೀತಾರೆ. ಎರಡನೇ ಲೇಯರ್ ಇಂಟೈನು, ಸೆಲ್ಯುಲೋಸ್ ಮತ್ತು ಪೆಕ್ಟಿನ್ ಆಗಿರ್ತದೆ. ಒಳಗಡೆ ಎರಡು ಸೆಲ್ ಇರ್ತವೆ, ಜನರೇಟಿವ್ ಮತ್ತು ವೆಜಿಟೇಟಿವ್ ಸೆಲ್. ವೆಜಿಟೇಟಿವ್ ಸೆಲ್ ಸೆಲ್ ವಾಲ್ ಏನಾಗಿರ್ತದೆ? ಇಂಟೈನ್ ಲೇಯರ್ ಆಗಿರ್ತದೆ. ಅದರಿಂದ ಏನು ಬರ್ತದೆ? ಪೋಲನ್ ಟ್ಯೂಬ್ ಬರ್ತದೆ. ಎಲ್ಲಿಂದ? ಪೋಲನ್ ಜರ್ಮ್ ಪೋರಿಂದ. ದೆನ್ ಸ್ಮಾಲರ್ ಸೆಲ್ ಯಾವುದು? ವೆಜಿಟೇಟಿವ್ ಸೆಲ್ ಆಯ್ತು. ಇನ್ನೊಂದು ಯಾವುದು? ಜನರೇಟಿವ್. ಅದು ಮೈಟೋಟಿಕ್ ಸೆಲ್ ಡಿವಿಷನ್ ಆಗಿ ಎರಡು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸನ್ನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾ ಬರ್ತದೆ. ಓಕೆ, ಇದಿಷ್ಟು ಪೋಲನ್ ಗ್ರೇನ್ ಬಗ್ಗೆ ಆಯ್ತು. ದೆನ್ ದೆರ್ ಇಸ್ ಅ ಪ್ರೊಸೆಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಾಲ್ ಇಟ್ ಆಸ್ ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಅಂತ ಬರ್ತದೆ. ಸೊ ಏನಪ್ಪ? ದ ಪ್ರೊಸೆಸ್ ಆಫ್ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೇನ್ ಫ್ರಮ್ ಪೋಲನ್ ಮದರ್ ಸೆಲ್. ಅಂದರೆ the pollen mother cell undergo meiotic cell division which form a 4 haploid functional pollen grain. 4 haploid functional pollen grains are going to be produced where when the process of microsporogenesis will complete. Then, so this is a microsporogenesis. Then another process we are going to call it as microgametogenesis. Once the pollen grains are formed, okay, it has a two cell, one is generative cell, second one is vegetative cell. The process of formation of pollen tube and the two male gamete inside the pollen grain we are going to call it as microgametogenesis. Okay, the process of formation of two male gametes and the pollen tube by the pollen grain we call it as microgametogenesis. Okay, microsporogenesis andre PMC ಇಂದ ಪೋಲನ್ ಮದರ್ ಸೆಲ್ ಇಂದ ಏನಾಗ್ತದೆ? ಪೋಲನ್ ಗ್ರೇನ್ ಬಂದರೆ ಅದು ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್. ಮೈಕ್ರೋಗ್ಯಾಮೆಟೋಜೆನೆಸಿಸ್ ಅಂದರೆ ಪೋಲನ್ ಗ್ರೇನಿಂದ ಪೋಲನ್ ಟ್ಯೂಬ್ ಬಂದು ಅದರಲ್ಲಿ ಎರಡು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮ್ ಆಗುವಂಥ ಪ್ರೊಸೆಸ್ಸನ್ನು ಮೈಕ್ರೋಗ್ಯಾಮೆಟೋಜೆನೆಸಿಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಕ್ಲಿಯರ್ ಆಯ್ತು ಅದು? ದೆನ್ ಇಲ್ಲೇನಾಗ್ತದೆ? ಆಫ್ಟರ್ ದ ಮೈಕ್ರೋಗ್ಯಾಮೆಟೋಜೆನೆಸಿಸ್ ದ ಜನರೇಟಿವ್ ಸೆಲ್ ಅಂಡರ್ಗೋ ಮೈಟೋಟಿಕ್ ಸೆಲ್ ಡಿವಿಷನ್. ಓಕೆ, ಮೈಟೋಟಿಕ್ ಸೆಲ್ ಡಿವಿಷನ್ ಟು ಫಾರ್ಮ್ ವಾಟ್? Two male gametes which are floating inside the pollen tube. So remember one important point: when the pollen tube headed towards the embryo sac, I can say the oval. So here it is led by, or it is the first nucleus we are going to observe at the tip of the pollen grain is the nucleus of vegetative cell. Secondly, or it is followed by the two male gametes, and these three nucleus are going to release at the time of when the pollen tube enter into the embryo sac. Okay, then then we move to some of the important concept regarding the pollen grain. So here pollen is said to be the two types. One is viable pollen grain, second one is non-viable pollen grain. So when I said to be viable pollen grains, those pollen grains which are going to show the growth of pollen tube on stigma, means those pollen grain are able to show the growth of pollen tube. ಯಾವೆಲ್ಲ pollen grain ಗೆ pollen tube grow ಮಾಡುವಂಥ ability ಇರ್ತದೆ, so they are said to be viable. Which do not have that property, they are called as non-viable pollen grain. You can say the viability of pollen grain, so that we have studied in a practical. So that is called as calculation of the percentage of pollen viability in a given flower in a second PUC practical exam. Okay, in the practical you have studied all these things. Okay, then so again when I talk about the duration of viability, remember some plants having the shorter time of viability period, whereas some having the years together. That means, so when I talk about the rice and wheat plant, okay, you can take a cereals, we are going to observe the viability of the pollen grains lies up to 30 minutes. Okay, beyond 30 minutes, the pollen grain cannot be in the state of viability. But some plant, for example, Rosaceae, Fabaceae, okay, so for like these families, we are going to observe the pollen viability may last up to months together, up to the years. ಕೆಲವು ತಿಂಗಳಿಂದ ವರ್ಷಗಟ್ಟಲೆ ಏನಾಗ್ತವೆ ಪೋಲನ್ ಗ್ರೀನ್ಸ್ ಹಾಗೆ ವಯಾಬಲ್ ಇರ್ತವೆ. ಸೊ ಇಲ್ಲಿಗೊಂದು ಎರಡು ಎಕ್ಸಾಂಪಲ್ಸ್ ಅದೇ. ಓಕೆ, ದಟ್ ಈಸ್ ಅ ಡೇಟ್ ಪಾಮ್ ಅಂತ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತಾರೆ. ಆಮೇಲೆ ಲುಪಿನಸ್ ಆರ್ಟಿಕುಲಸ್ ಅಂತ ಒಂದು ಪ್ಲಾಂಟ್ ಅದೇ. ಸೊ ಹಿಯರ್ ಇನ್ ದೀಸ್ ಕೇಸಸ್ ವಾಟ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಅಬ್ಸರ್ವ್ ದ ಪೋಲನ್ ವ್ಯಾಬಿಲಿಟಿ ಮೇ ಲಾಸ್ಟ್ ಅಪ್ ಟು 10,000 ಇಯರ್ಸ್. ಓಕೆ, ಸಾವಿರಾರು ವರ್ಷಗಟ್ಟಲೆ ಸಹಿತ ಏನಾಗ್ತದೆ ಪೋಲನ್ ಗ್ರೇನು ಹಾಗೆ ವಯಾಬಲ್ ಇರ್ತಾ ಹೋಗ್ತದೆ. ಓಕೆ, ದೆನ್ ಇದಾದ್ಮೇಲೆ ಒಂದು ಪಾಯಿಂಟ್ ಹೇಳ್ತಾರೆ, ದ ಪೋಲನ್ ಆಲ್ಸೋ ಹ್ಯಾವಿಂಗ್ ಟು ಅಡ್ವಾಂಟೇಜಸ್, ಸಾರಿ ಒನ್ ಅಡ್ವಾಂಟೇಜ್ ಆ್ಯಂಡ್ ಒನ್ ಡಿಸ್ಅಡ್ವಾಂಟೇಜಸ್, ದಟ್ ಈಸ್ ವಿ ಕಾಲ್ ಎಕನಾಮಿಕ್ ಇಂಪಾರ್ಟೆನ್ಸ್. When I talk about pollen beneficial aspect, so here it is act as the syrup, that is pollen syrup is prepared from the pollen grain, which is used to increase the stamina in horses as well as in case of the athletes. Okay, those are involved in the athletics as well as the race horses, they are fed up with these pollen grains. Okay, the pollen syrup or in the form of pollen tablet, they are going to increase the stamina. ಅದೇನು ಮಾಡ್ತದೆ, ಸ್ಟೆಮಿನಾವನ್ನು ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ತದೆ. ಅಟ್ ದ ಸೇಮ್ ಟೈಮ್ ದ ಪೋಲನ್ ಗ್ರೇನ್ ಮೇ ಆಲ್ಸೋ ಕಾಸ್, okay, the pollen grain may also cause somewhat the harmful effect. The major problem is we are going to observe what the pollen allergy. It may lead to the bronchitis, the asthma, or the severe respiratory disorders. Okay, this is about what the male part. When I come for female part, so here the first point you have to remember the structure of ovule. That is we call it as megasporangium. The microsporangium nothing but your anther, and megasporangium nothing but the ovule. When I talk about ovule, the ovule is attached to the wall of ovary. Okay, the ovule is attached to the wall of ovary with the help of a connection, we call it as placenta. Then the placenta and the ovule, which is attached by a stalk-like structure, we call it as the funicle. Okay, funicle is a stalk-like structure which helps in attachment of body of ovule to the placenta. Placenta is a cushion-like structure which get connected from the wall of the ovary and the developing ovule. Then again, the body of an ovule is having, I can say, the junction between the body of an ovule and the funicle, we call it as hilum. Then body of an ovule is surrounded by two wall layers, you can say two layer, that is we call it as integument, outer integument and inner integument. So here at the tip of the ovule where the integuments are absent, there is a formation of a small pore, we call it as micropyle. So that region we are going to call it as micropylar region. At the base of a ovule we find a region called as chalazal. Okay, here the ovule has two ends, one is micropylar end where the micropyle is present. Second one we are going to call it as what, the chalazal end where the chalaza is present. Then a central core tissue present at the center of an ovule, we call it as what, the nucellus. So that is a nutritive tissue. Okay, that is a nutritive tissue which plays a very important role in nourishment of developing embryo sac. At the center of each ovule we find one embryo sac. So that is made up of the 7 cell 8 nucleated structure. That is about ovule. So here there are different types of ovule. Okay, that is we generally we are going to observe anatropous ovule, which is the body of an funicle and the body of an ovule and the funicle which are 180 degree. Okay, the body is turned to 180 degree where they lies in the same parallel lines in an inverted fashion. Is it clear? Then there are different type. Anatropous ovule is there, that is about 180 degree. So hemianatropous, that is 90 degree. Arthropous lies in the same straight line. So these are the different types of ovule. Then come to the procedure. So what is that? Megasporogenesis. So here the tricky point I would remember in megasporogenesis is, so when I say megasporogenesis, please remember when I say megasporogenesis, what the thing you have to remember is, after completion of the one megasporogenesis, in generally we are going to observe the 4 haploid megaspore. Okay, the process is again same, formation of megaspore from megaspore mother cell, no doubt in that. So at the end of one megasporogenesis there is a formation of 4 haploid ಮೆಗಾಸ್ಪೋರ್. ಇಲ್ಲಿ ಏನಾಗ್ತವೆ, 4 ಹೆಪ್ಲಾಯ್ಡ್ ಮೆಗಾಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತವೆ. ಸೊ ಹಿಯರ್ the megaspore which is nearer to the chalazal end, please remember this point, the megaspore which is nearer to the chalazal end get converted into functional megaspore, and remaining 3 which are below that functional megaspore, they get converted into degenerated. Okay, they become non-functional. So you can remember a trick here, first from chalazal or the last from the micropylar. Okay, you can say first cell from the chalazal or the last cell from the micropylar end, they are going to form a functional megaspore, that is called as megasporogenesis. Then megagametogenesis, once the functional megaspore is formed, so from that functional megaspore the embryo sac going to develop. Okay, the embryo sac will going to develop. That process we are going to call it as megagametogenesis. It is called as megagametogenesis. So here when I say megagametogenesis, the functional megaspore undergo 3 free nuclear division. So when I say 3 free nuclear division, please remember here 3 free nuclear division antandre the nucleus will going to divide, and here that process is not accompanied by the formation of a cell wall, only the karyokinesis will going to occur. ಓಕೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ಮಾತ್ರ ಡಿವೈಡ್ ಆಗ್ತದೆ, ಅಲ್ಲಿ ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ಆಗಲ್ಲ. ಆ ಥರ ಡಿವಿಷನ್ ಏನಂತ ಕರೀತಾರೆ, ಫ್ರೀ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ when I say free nuclear division, please remember here. Okay, so ಇಲ್ಲಿ ಏನಾಗ್ತದೆ, at the end of 3 free nuclear division there is a formation of 8 nucleus and they are going to rearrange. Okay, they are going to rearrange to form a 7-celled 8 nucleated structure. So here when there is a first free nuclear division, there is a formation of 2 nucleated embryo sac, second 4 nucleated embryo sac, third 8 nucleated embryo sac, and 1 nucleus from the chalazal as well as micropylar, they come to the center, they form a polar nucleus. Then the 3 which are present to the micropylar end, they get converted into egg apparatus, which consisting of 2 synergid and 1 egg cell, and the 3 nucleus present at the chalazal end, they get converted into antipodals. So here I'm going to talk about the matured embryo sac. Please remember, matured embryo sac, it having a 7-celled 8 nucleated, 3 are the chalazal, 3 are the antipoda. Okay, that is antipodals. Okay, 3 are the chalazal, that is called as antipodals, and 3 are the micropylar, that are called as egg apparatus consisting of an egg cell and 2 synergid. And here these are enclosed in a bigger cell, that is called as central cell. So totally 7 cell. Centrally we are going to observe 2 nucleus plus 3 plus 3, there are 8 nucleus, hence called as 8 nucleated 7-cell structure. Okay, at the synergid we find a finger-like projection, they are called as what? Filiform apparatus. So they are going to secrete certain chemical which guide the entry of pollen into the embryo sac. This is about the female part. Then come to the pollination, the process of transfer of anther from the anther to the stigma, the process of transfer of pollen from anther to the stigma. So here the pollination of 2 different types. So one we call it as a self pollination, second one we call it as cross pollination. So generally when I go with the self pollination, again we are going to divide into 2 types. One is autogamy and second one zetnogamy. So when I say zetnogamy and autogamy, both are going to represent the self pollination, and second one we are going to call it as xenogamy, that are going to represent what? The cross pollination. So when I say autogamy, remember the trick here, a pollination occur within a single flower. Okay, if the pollination occur within a same flower, that is called as autogamy. If it is between the 2 different flowers of a same plant, which going to remembered as within a flower, we call it as autogamy. 2 different flowers of a same plant, that is called as zetnogamy. So here in the zetnogamy, remember one important quote here. So here it bring genetically similar pollen grains. Zetnogamy result in a bringing of genetically similar pollen grain and seems to be a cross pollination. ನೋಡ್ಲಿಕ್ಕೆ ಅದು ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಅನಿಸ್ತದೆ, ಬಟ್ ಅದೇನಾಗ್ತದೆ ಜೆನೆಟಿಕಲಿ ಸಿಮಿಲರ್ ಪೋಲನ್ ಗ್ರೈನ್ಸ್ ಅನ್ನು ತಗೊಂಡು ಬರೋ ಉದ್ದೇಶಕ್ಕೆ ಏನು ಮಾಡ್ತದೆ, ಸೊ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಅ ಝೀಟೋನೋಗ್ಯಾಮಿ. ವೆನ್ ಐ ಕಮ್ ಫಾರ್ ಝೀನೋಗ್ಯಾಮಿ, ಸೊ ರಿಮೆಂಬರ್ ಹಿಯರ್ in case of xenogamy, so here it is completely known as the cross pollination. The pollination occurs between two flowers of two different plant. Okay, between the two flowers of two different plant, we call it as cross pollination or xenogamy. Then so here the flowers are said to be two types here. One we call it as what, chasmogamous flower, and second one we are going to call it as clistogamous flower. So chasmogamous nothing but the flower which is said to be opened flower or well exposed flower. Similarly, when I talk about the clistogamous flower, they are also called as closed flower which do not open at all. For example, oxalis is there, commelina is there, viola is there. So they are going to exhibit the closed flower. They are also called as clistogamous. Then you can ask an advantage. So when I say if the flower is clistogamous, there are certain advantage. Number one, it can always undergo autogamy, or you can say self-pollination. Second one, helps to maintain homozygous condition, that is pure line. Third one, not having any dependency on, or you can say no dependency on the pollinator. Okay, pollinator ಮೇಲೆ ಡಿಪೆಂಡ್ ಆಗುವಂಥ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಸೊ ಇವೇನು ಕ್ಲಿಸ್ಟೋಗ್ಯಾಮಸ್ ಫ್ಲವರ್ದು ಇಂಪಾರ್ಟೆಂಟು ಅಡ್ವಾಂಟೇಜಸ್ ಅಂತ ಇದನ್ನು ನಾವು ಕರಿತಾ ಹೋಗ್ತೇವೆ. ಓಕೆ, ಇದಾದಮೇಲೆ ಮುಂದೆ, ಸೊ ಮುಂದೆ when I talk about pollination, if it is cross pollination, or you can say if it is zetonogamy, so in case of autogamy there is no requirement of any agent. But here when I come for zetonogamy or when I come for xenogamy or cross pollination, so here the plant requires certain agents. So they are called as, we call it as a pollinating agents, we can say pollinators. So here in some plant that depends upon the wind, or you can say air, they are called as anemophilous flower or wind pollinating flower. So similarly when I talk about second one, that is some are, they are pollinated by water, they are called as hydrophilous flower, we can say hydrophily. And the third one we call it as insect or entomophily ಅಂತ ಕರೀತಾರೆ. ಇವು ಮೂರನ್ನು ನೆನ್ಪಿಟ್ಕೊಟ್ರು. ಈಗಪ್ಪ ಎಂಥ ಫ್ಲವರ್ ಒಳಗಡೆ ಅನಿಮೋಫಿಲಸ್ ಇರ್ತದೆ ಅಂದರೆ, ದ ಫಸ್ಟ್ ಪಾಯಿಂಟ್, ದ ಫ್ಲವರ್ ಶುಡ್ ಬಿ ವೆಲ್ ಎಕ್ಸ್ಪೋಸ್ಡ್. ಓಕೆ, ಕಂಪ್ಲೀಟಾಗಿ ಓಪನ್ ಆಗಿರಬೇಕು, ಎಲ್ಲ ಪಾರ್ಟ್ಸು ಗಾಳಿಯಲ್ಲಿ ತೆರೆದಿರಬೇಕು, ಎಕ್ಸ್ಪೋಸ್ ಆಗಿರಬೇಕು. Second one, the pollen grain should be light, non-sticky, and they have to produce huge number of pollen grain in order to the carry from the wind. Similarly, the stigma should be feathery in order to trap the pollen grain from the air. Similarly, each ovary consisting of only one ovule, that is the characteristics of what, wind pollinating flower. Similarly, when I talk about insect pollinating flower, the flower should be brightly colored, where in case of wind there is no production of nectar and there is no colored flowers there. But here in case of the entomophilus, the important condition is the flower should be colored, okay, then has to produce nectar. Sometimes they are having a scented odor. Okay, similarly, so the pollen grains are sticky. Okay, pollen grains are sticky in nature, and the stigma is also sticky in nature in order to adhere the pollen grain on the surface. Okay, these are some important character of the insect and wind pollinating flower. Then when I come for anemophilous flower, hydrophilous flower, again there are two types. Okay, remember number one, the first one we call it as what, the epihydrophilous flower. Second one, hypohydrophilous. That means if the pollination occurs on the surface of water, for example, we call it as Vallisneria. So what happens in Vallisneria? Both male and female flower come on the surface. Okay, then male flower release the pollen grains on the surface of water. They passively bring towards the female flower, that bring about the pollination. Similarly, in case of the hypohydrophily, so what happens here? Both male and female flowers present inside the water. The pollen grains release in the form of a ribbon or chain-like, and this will reach to the female flower, that bring about the pollination. Okay, then all these things once it has happened. Okay, remember the two examples here. One is the Vallisneria. This is the example for epihydrophilia, or the pollination occurs on the surface of the water. Similarly, hypohydrophily. Okay, when I talk about similarly hypohydrophily, it is also a condition where the pollination occurs inside the water. So this is about what the pollinators. Okay, ಇವೆಲ್ಲ ಏನಾಯಿತು pollinators ಬಗ್ಗೆ ಆಗ್ತದೆ. Then ಇದಾದ್ಮೇಲೆ ಮುಂದೆ we go for an important process that is called as outbreeding devices. Means what are the factors which always prevent the self pollination in a flower, that is we call it as outbreeding devices or the contrivances of the cross pollination. Number one, dichogamy. So what do you mean by dichogamy? If both anther and stigma matures the same time, both anther dichogamy means anther and stigma matures at a different time, because one day the anther will mature and another day stigma will mature. So here there is no synchronization. Okay, there is no similarity, there is no synchronization. If there is no synchronization, then there is no chance of self pollination. Okay, then second one, dicliny. The flower should be unisexual. If the flower is unisexual, then it always in favor of what? Cross pollination. Number three, we call it as heterostyly. So remember, in some flower the length of the style varies with the length of stigma. Okay, the length of style is going to vary from flower to flower. Some flower they having very shorter style, and in some flower we are going to observe the elongated style. The shorter and the elongated style with respect to the length of the filament, if it is in different length, then there is also no chance for any cross pollination. Okay, then next what we are going to observe here, next we call it as hercogamy. So here both anther and stigma matures in the same time, but still there is a physical barrier present between the anther and the stigma which cannot allow any transfer of pollen grains, hence it is going to prevent a self pollination. And lastly, we call it as self sterility. Remember, if the pollen grains are produced from the same flower having same genetic constitution, they are going to rejected by, okay, if the pollen grain and the stigma having the same gene, okay, same similarity, same relationship, then they are going to be rejected by an important phenomenon we call it as self sterility. So here the self sterility is a genetic factor. ಅದು ಏನಂತ ಕರೀತಾರೆ? Genetic factor ಅಂತೇಳಿ ಕರೀತಾರೆ. The next one, so we'll go with the pollen pistil interaction ಅಂತ ಬರ್ತದೆ. ಸೊ ಏನಪ್ಪ pollen pistil interaction ಅಂದರೆ, ಸೊ ಇಲ್ಲಿವರೆಗೂ ನಾವು ಪಾಲಿನೇಷನ್ ನೋಡಿದ್ವಿ, ಸೆಲ್ ಪಾಲಿನೇಷನ್ ಎಲ್ಲ ಇವೆಂಟ್ಸ್ ಅನ್ನು ನೋಡಿದ್ವಿ. ಏನಾಗ್ತದೆ ಆ್ಯಕ್ಚುಲಿ ಪೋಲನ್ ಗ್ರೇನು ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ ಬಿದ್ದಾಗ, ಸೊ ಏನಾಗ್ತದೆ ಅನ್ನೋದು ಎಲ್ಲ ಇವೆಂಟ್ಸ್ ಅನ್ನು ಸರಿ ಏನು ಮಾಡ್ತಾರೆ, ಪೋಲನ್ ಪಿಸ್ಟಲ್, ಪೋಲನ್ ಅಂದರೆ ಪೋಲನ್ ಗ್ರೇನ್ಸು, ಪಿಸ್ಟಲ್ ಅಂತಂದರೆ ಫೀಮೇಲ್ ಪಾರ್ಟು, ಯಾವ ರೀತಿ ಇಂಟ್ರಾಕ್ಷನ್ ಆಗ್ತದೆ. ಸೊ ಹಿಯರ್ ಇಟ್ ಇಸ್ ಅ ಸಾರ್ಟ್ ಆಫ್ ಅ ಕೆಮಿಕಲ್ ಇಂಟ್ರಾಕ್ಷನ್ ಆರ್ ಇಂಟ್ರಾಕ್ಷನ್ ಬಿಟ್ವೀನ್ ದ ಕೆಮಿಕಲ್ ಸಿಕ್ರೇಷನ್. ಸೊ ಇದೇನೋ ಒಂದು ಸೆಕ್ರೇಟ್ ಮಾಡ್ತದೆ, ಅದೊಂದು ಯಾವ ರೀತಿ ರೆಸ್ಪಾಂಡ್ ಮಾಡ್ತದೆ ಅನ್ನೋದು ಇಂಟ್ರಾಕ್ಷನ್, ದಟ್ ಇಸ್ ಅ ಕಮ್ಯುನಿಕೇಶನ್ ಬಿಟ್ವೀನ್ ದ ಪೋಲನ್ ಗ್ರೇನ್ ಆ್ಯಂಡ್ ಸ್ಟಿಗ್ಮಾ. ದಟ್ ಇಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಾಲ್ ಇಟ್ ಆಸ್ ವಾಟ್, ಅ ಪೋಲನ್ ಪಿಸ್ಟಲ್ interaction. So here in the pollen pistil interaction, so remember in the pollen pistil interaction there are different steps. Step number one, when the pollen grain fall upon the stigma, that process we are going to call it as a pollination. The first one is pollination. Once a pollination occur, we know that, so if it is a wind pollination or insect pollination, it may bring the undesirable pollen grain from a different plant also. Means the pollen grain which are fall upon the stigma, there is no guarantee that all the pollen grain belong to that plant. It may have some undesirable pollen grains also. So that is why, remember, that is why, so we are going to observe there is acceptance or rejection of the pollen grain by the stigma. ಸ್ಟಿಗ್ಮಾ ಏನು ಮಾಡ್ತದೆ, ಪೋಲನ್ ಗ್ರೇನ್ ಅಕ್ಸೆಪ್ಟ್ ಮಾಡ್ಬೋದು ಅಥವಾ ರಿಜೆಕ್ಟ್ ಮಾಡುವಂಥ ಎಬಿಲಿಟಿ ಇರ್ತದೆ. ಸೊ ಹೀಗಾಗಲಿ ಏನಾಗ್ತದೆ, ನೆನ್ಪಿಟ್ಕೊಡ್ರಿ, ಎರಡನೇ ಸ್ಟೆಪ್ ಏನು, ಅಕ್ಸೆಪ್ಟೆನ್ಸ್ ಆರ್ ರಿಜೆಕ್ಷನ್ ಆಫ್ ದ pollen grain. Number 3, so because of accepted pollen grain, then the stigma will secrete a stigmatic fluid. So that stigmatic fluid consisting of sugars, the boric acid, the magnesium and nitrate content, which trigger the growth of pollen tube on the stigma. ಅಲ್ಲಿ ಏನಾಗ್ತದೆ, pollen grains pollen tube ಅನ್ನು ಗ್ರೋ ಮಾಡಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತವೆ. ಯಾವಾಗ, once they are going to be accepted and the stigma has to secrete what, a stigmatic fluid. Then the germinated pollen grain slowly, what happens here, invade the stigma, then style ultimately reaches to the oval. Oval ಅನ್ನ ಎಂಟ್ರಿ ಮಾಡ್ತದೆ. ಇಷ್ಟು ಪ್ರೊಸೀಜರನ್ನು ಏನಂತ ಕರೀತಾರೆ? ಸೊ ಜನರಲಿ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಾಲ್ ಇಟ್ ಆಸ್ ವಾಟ್? The pollen pistil interaction. For a competitive examination, remember the 3 point: pollination, acceptance or the rejection of the pollen grain, and third one, the growth of pollen tube until it enters into the ovary. Sorry, that is ovule. So all these processes we are going to call it as a pollen pistil interaction. By seeing these mechanism, the human being going to modify the pollination process by using the desirable pollen grain, that is we are going to call it as artificial pollination. There are different steps there. So what is the first one? We go for an important step called as emasculation. So what is emasculation? E refers to removal, masculation refers to the male part. So it is nothing but removal of a male part from a bisexual flower without disturbing the female part, that is we are going to call it as what? The emasculation. The second one, so before going to the second one, you have to remember some of the important criteria for emasculation. The emasculation can be done only when the flower is at bud condition, and that bud should be opened in a next day, or you can say it should be in opening in a next day. That is called as what? Pollen. So here emasculation can be done when the flower is supposed to open in ನೆಕ್ಸ್ಟ್ ಡೇ ಮಾರ್ನಿಂಗ್, ನಾಳೆ ಬೆಳಗ್ಗೆ ಓಪನ್ ಆಗುವಂಥ ಫ್ಲವರ್ ಒಳಗಡೆ ಏನು ಮಾಡ್ತಾರೆ? ಇಮ್ಯಾಸ್ಕುಲೇಷನ್. ಇದು ಒಂದು ಪಾಯಿಂಟ್ನ ಅಬ್ಸರ್ವ್ ಮಾಡ್ತಾರೆ. ಸೆಕೆಂಡ್ ಒನ್, ದೇರ್ ಈಸ್ ನೋ ಆಂಥಸಿಸ್. ಓಕೆ, ಮೀನ್ಸ್ ದೇರ್ ಈಸ್ ನೋ ಡಿಹೈಸನ್ಸ್ ಆಫ್ ಪೋಲನ್ ಗ್ರೇನ್ ಇನ್ಸೈಡ್ ದ ಬಡ್. ಸೊ ಒಂದು ಬಡ್ಡನ್ನು ಓಪನ್ ಮಾಡಿದರೆ ಪೌಡ್ರಿ ಮಾಸ್ ಇತ್ತು, ಓಕೆ, ಆಂಥರ್ ಡಿಹೈಸ್ ಆಗೈತೆ ಅಂದರೆ ದ್ಯಾಟ್ ಫ್ಲವರ್ ಕೆನ್ ನಾಟ್ ಬಿ ಟೇಕನ್ ಫಾರ್ ಎಮ್ಯಾಸ್ಕುಲೇಷನ್. Then emasculation can be done with a different method. Some flower we can go with the hand emasculation by using force. We can remove the anther. In some flower, if the flower is very small, then it can be treated with acid or what? The hot water, in order to make the anther to be a sterile one. So these are the different methodology we can use for emasculation. And remember, very important point, in case of unisexual female flower, there is no need of emasculation. Then second one, bagging. So what happens here? The emasculated flower has to be covered with what? The butter paper bag, making a small pores on it in order to avoid the undesirable pollen grain which may fall upon the stigma. ಸೊ ಬೇರೆ pollen grain ಬೀಳಾರ್ದಂಗೆ ಏನು ಮಾಡ್ಬೇಕು? ನಾವು ಅದನ್ನು ಕವರ್ ಮಾಡಬೇಕು. ಆ ಪ್ರೊಸೆಸ್ಗೆ ಬ್ಯಾಗಿಂಗ್ ಅಂತ ಕರೀತಾರೆ. ದೆನ್ ವಿ ಗೋ ಫಾರ್ ಆರ್ಟಿಫಿಷಿಯಲ್ ಪಾಲಿನೇಷನ್. ನೆಕ್ಸ್ಟ್ ಮಾರ್ನಿಂಗ್ ಏನು ಮಾಡ್ತಾರೆ? ಆ ಬ್ಯಾಗನ್ನು ತೆಗೆದು ಡಿಸೈರಬಲ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಅಂತ ಯಾವ ಕ್ಯಾರೆಕ್ಟ್ನು ಆ ಪ್ಲಾಂಟಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಬೇಕು, ಆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ನಲ್ಲಿ ಏನು ಮಾಡ್ತಾರೆ? ಡಸ್ಟ್ ಮಾಡ್ತಾರೆ. ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಆರ್ಟಿಫಿಷಿಯಲ್ ಪಾಲಿನೇಷನ್. ಅಗೇನ್ ದಿ ಗೋ ಫಾರ್ ರೀಬ್ಯಾಗಿಂಗ್. ಮತ್ತೆ ಅದನ್ನು ಹೊಳ್ಳಿ ಬ್ಯಾಗ್ ಮಾಡ್ತಾರೆ, ಬ್ಯಾಗಿಂಗನ್ನು ಮಾಡ್ತಾರೆ, ಅದಕ್ಕೆ ರೀಬ್ಯಾಗಿಂಗ್ ಅಂತ ಕರೀತಾರೆ. Then after the rebagging, there is a formation of fruit and seed. Then they are going to harvest the seed. These are artificial pollination we are going to observe in case of what? The flowering plants. Then come to the fertilization. So already we are having an awareness that when I say fertilization, there is a fusion of male and female gamete. Since in angiosperm, so we are going to observe double fertilization and triple fusion. When I say double fertilization, triple fusion, please remember, so when I say double fertilization, that means there are two fertilization we are going to observe. Means there is a fusion of a different nucleus there. ಎರಡು ಬೇರೆ ಬೇರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಲ್ಲಿ ಫ್ಯೂಸ್ ಆಗೋದನ್ನು ಅಬ್ಸರ್ವ್ ಮಾಡ್ತೇವೆ. ಸೊ ಟ್ರಿಪಲ್ ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಏನು? ಒಂದು ಫ್ಯೂಷನ್ ಒಳಗಡೆ ಮೂರು ನ್ಯೂಕ್ಲಿಯಸ್ ಫ್ಯೂಸ್ ಆಗ್ತವೆ. ಸೊ ಹಿಂಗಾಗಿ ಅದನ್ನು ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಆ್ಯಂಡ್ ಟ್ರಿಪಲ್ ಫ್ಯೂಷನ್. ಈಗ ಫಸ್ಟು ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಬಗ್ಗೆ ನೋಡ್ತಾ ಬರೋಣ. ಸೊ ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂದ್ರೆ ಏನಾಗ್ತದೆ? ಸೊ here remember, in the double fertilization, the pollen grain carries two male gametes. ಆಲ್ರೆಡಿ ಗೊತ್ತದು, pollen grain ಒಳಗಡೆ ಎರಡು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಬರ್ತಾ ಇರ್ತವೆ. ಆ ಎರಡು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಏನಾಗ್ತದೆ? ಒಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟು ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಜೊತೆ ಫ್ಯೂಸ್ ಆಗಿ ಏನು ಮಾಡ್ತದೆ? ಒಂದು ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗ್ತದೆ. ಇನ್ನೊಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟು ಎಗ್ ಸೆಲ್ ಜೊತೆ ಫ್ಯೂಸ್ ಆಗಿ ಇನ್ನೊಂದು ಫರ್ಟಿಲೈಸೇಷನ್ ಅನ್ನ ಮಾಡ್ತದೆ. ಹಾಗಾಗಿ ಎಷ್ಟು ಫರ್ಟಿಲೈಸೇಷನ್ ಆಯ್ತು ಈಗ? ಎರಡು. ಹೆನ್ಸ್ ಇಟ್ ಈಸ್ ಡಬಲ್ ಫರ್ಟಿಲೈಸೇಷನ್. ದ ಫಸ್ಟ್ ಫರ್ಟಿಲೈಸೇಷನ್, the male gamete fuses, they get converted into zygote. Zygote ಕನ್ವರ್ಟ್ ಆಗ್ತದೆ. ಎರಡನೇ ಫರ್ಟಿಲೈಸೇಷನ್ ಯಾವುದು? the male gamete fuses the polar nucleus, get converted into what? the primary endosperm nucleus. that is generally we call it as pen ಅಂತ ಕರೀತಾರೆ. primary endosperm nucleus ಅಂತ ಕರೀತಾರೆ. ಇದಿಷ್ಟು ನೆನ್ಪಿಟ್ಕೊಡ್ರಿ. then why it is called a triple fusion? look at the second fertilization or the formation of pen, primary endosperm nucleus. what happens here? primary endosperm nucleus here, remember, ನೆನ್ಪಿಟ್ಕೊಡ್ರಿ, ಏನಾಗ್ತದೆ? ಎರಡು ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಅದೇ ಒಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಬಂತು ಫ್ಯೂಸ್ ಆಗ್ತದೆ, ಹೆನ್ಸ್ ದೆರ್ ಇಸ್ ಅ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಅ ಟ್ರಿಪ್ಲಾಯ್ಡ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಝೈಗೋಟ್ ಏನಿರ್ತದೆ, ಡಿಪ್ಲಾಯ್ಡ್ 2 ಏನಿದ್ರೆ ಎಂಡೋಸ್ಪರ್ಮ್ ಏನಿರ್ತದೆ ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಕಂಡೀಷನ್ದಲ್ಲಿ ಇರ್ತದೆ. ಇವಿಷ್ಟನ್ನು ಅಲ್ಲಿ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, ಅದು ಫರ್ಟಿಲೈಸೇಷನ್ ಇವೆಂಟಲ್ಲಿ. ಓಕೆ, ದೆನ್ ಇದಾದಮೇಲೆ ಈಗ ಕೆಲವೊಂದು ಡಿಫ್ರೆನ್ಸನ್ನು ಅಬ್ಸರ್ವ್ ಮಾಡ್ತಾ ಬರ್ತಾನೆ. ವಾಟ್ ಆರ್ ದ ಚೇಂಜಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಅಬ್ಸರ್ವ್ ಆಫ್ಟರ್ ದ ಫರ್ಟಿಲೈಸೇಷನ್ ಇನ್ ಅ ಫ್ಲವರ್? ಸೊ ಪೆಟಲ್ಸ್ ಫಾಲೋ ಅಪ್ ಆಗ್ತವೆ, ದೆನ್ ಅಲ್ಲಿ ಎಕ್ಸಲ್ ಇದು ಏನಾಗ್ತದೆ ಝೈಗೋಟ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ತದೆ, ದೆನ್ ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಏನಾಗ್ತದೆ ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್, ಮುಂದೆ ಅದು ಏನಾಗ್ತದೆ ಎಂಡೋಸ್ಪರ್ಮ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ತದೆ, ಈ ಝೈಗೋಟ್ ಮುಂದೆ ಎಂಬ್ರಿಯೋ ಆಗ್ತದೆ. ಸೊ ಇವೆಲ್ಲ ನೆನ್ಪಿಡ್ತಾ ಹೋಗ್ಬೇಕು. ಓಕೆ, ಇದಿಷ್ಟು. ದೆನ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಅಂದರೆ ಏನೀಗ? ಸೊ ಎಂಡೋಸ್ಪರ್ಮ್ ಇಸ್ ಅ ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಟಿಶ್ಯೂ, 3N ಇರ್ತದೆ. ಓಕೆ, ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಟಿಶ್ಯೂ ವಿಚ್ formed by the fusion of two polar nucleus and one male gamete during a fertilization. Then what is the function? The endosperm plays a very important role in the nourishment of developing embryo in the seed. And here, based on the presence and absence of endosperm, the seeds are categorized into 2 types. One is non-endospermic seed, where the endosperm is there but it is completely consumed when the seed become matured. Second one, in case of all the poaceae member, or in case of the grasses, or in case of cereals, we are going to observe the endosperm is present until the complete maturation of the seed, and it is also present during the germination. They are called as endospermic seed or albuminous seed ಅಂತನೂ ಕರೀತಾರೆ. ಓಕೆ, ದೆನ್ ಇದಾದ್ಮೇಲೆ ಸೊ ಮುಂದೆ ಏನಾಯ್ತು ಈಗ, ನೆಕ್ಸ್ಟ್ ವಿಲ್ ಗೋ ಫಾರ್ ಅನದರ್ ಇಂಪಾರ್ಟೆಂಟ್ ಕಾನ್ಸೆಪ್ಟ್, ದಟ್ ಈಸ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಎಂಬ್ರಿಯೋ. ಸೊ ಎಂಬ್ರಿಯೋ ಹೆಂಗೆ ಡೆವಲಪ್ ಆಗ್ತದೆ? So here there are different stages of development of embryo. So you remember zygote is a single cell which undergo first cleavage which form a pro-embryo. ಇಲ್ಲಿ ಏನಾಗ್ತದೆ, ಎಂಬ್ರಿಯೋ ಇದ್ದಿದ್ದು ಝೈಗೋಟ್ ಇದ್ದು ಡಿವೈಡ್ ಆಗಿ 2 ಸೆಲ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತದೆ. ಆ 2 ಸೆಲ್ ಅನ್ನ ಏನಂತ ಕರೀತಾರೆ, ಪ್ರೋ ಎಂಬ್ರಿಯೋ ಅಂತ ಕರೀತಾರೆ. ಸೊ ಹಿಯರ್ ದ ಬೇಸಲ್ ಸೆಲ್, ಕೆಳಗಡೆ ಇರುವಂಥ ಸೆಲ್ ಮತ್ತೆ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗಿ ಏನಾಗ್ತದೆ, ಇಟ್ ಫಾರ್ಮ್ಸ್ ಅ ಗ್ಲೋಬುಲಾರ್ ಶೇಪ್ಡ್ ಎಂಬ್ರಿಯೋ, ದೆನ್ heart shape embryo, and finally get converted into what? The matured embryo. Stages ಅನ್ನು ನೆನ್ಪಿಟ್ಕೊಂಡು ಕೆಲವು ಕ್ವಶನ್ ಕೇಳ್ತಾನೆ. ಒಂದಿಂದ ಎಲ್ಲಿಗೆ ಝೈಗೋಟ್, ಪ್ರೋ ಎಂಬ್ರಿಯೋ, ಅದಾದಮೇಲೆ ಗ್ಲೋಬುಲಾರ್ ಎಂಬ್ರಿಯೋ, ಹಾರ್ಟ್ ಶೇಪ್ ಎಂಬ್ರಿಯೋ, ಆ್ಯಂಡ್ ಫೈನಲಿ ವಾಟ್? ದ ಮ್ಯಾಚ್ಯೂರ್ಡ್ ಎಂಬ್ರಿಯೋ. ಇವಿಷ್ಟು ಸ್ಟೇಜಸ್ ಅಲ್ಲಿ ಅದ. ಓಕೆ, ಇದಿಷ್ಟು ಅರ್ಥ ಮಾಡ್ಕೊಡ್ರಿ. ದೆನ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಎಂಬ್ರಿಯೋ ನೋಡೋದಾದ್ರೆ 2 ಥರ ಎಂಬ್ರಿಯೋ ಅಬ್ಸರ್ವ್ ಮಾಡ್ತೇವೆ. ಡೈಕಾಟ್ ಎಂಬ್ರಿಯೋ ಬೇರೆ, ಮೊನೋಕಾಟ್ ಎಂಬ್ರಿಯೋ ಬೇರೆ. ಸೊ ಇಲ್ಲೇನು ಎಂಬ್ರಿಯೋನಿಕ್ ಆಕ್ಸಸ್ ನೆನ್ಪಿಟ್ಕೊಡ್ರಿ. ಅಪ್ಪರ್ ಪಾರ್ಟನ್ನು ಫಿನಿಕಲ್ ಏನಂತ ಕರೀತಾರೆ, ಓಕೆ, ಬೇಸಲ್ ಪಾರ್ಟನ್ನು ರ್ಯಾಡಿಕಲ್ ಅಂತ ಕರೆದ್ರೆ, ಅಪ್ಪರ್ ಪಾರ್ಟನ್ನು ಶೂಟ್ ಪಾರ್ಟ್ ಅಂತ ಕರೀತಾರೆ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ಕೋಲಿಯೋಪ್ಟಾಯಿಲ್ ಅಂತ ಬರ್ತದೆ, ಕೋಲಿಯೋರೈಸಾ ಅಂತೇಳಿ ಬರ್ತದೆ, ಮೊನೋಕಾಟಲ್ ಬರ್ತದೆ, ಡೈಕಾಟಲ್ ಬಂದಾಗ ಎಪಿಕೋಟಾಯಿಲ್, ಹೈಪೋಕೋಟಾಯಿಲ್ ಅಂತ ಬರ್ತದೆ. ಸೊ ಇವಿಷ್ಟು ಬೇರೆ ಬೇರೆ ಪಾರ್ಟ್ಸನ್ನು ನೀವು ನೆನ್ಪಿಡಬೇಕು. ಓಕೆ, ಎಪಿಕೋಟೈಲ್, ಹೈಪೋಕೋಟೈಲ್ ಅಲ್ಲಿ ಬರ್ತದೆ ಇನ್ ಕೇಸ್ ಆಫ್ ಡೈಕಾಟ್. ಅದೇ ನೀವು ಮೊನೋಕಾಟಲ್ ಬಂದರೆ ದ ಎಂಬ್ರಿಯೋ ಇಸ್ ಕವರ್ಡ್ ಬೈ ಎ ಸ್ಪೆಷಲೈಸ್ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಪ್ಲುಮಿಲ್ ಇಸ್ ಕವರ್ಡ್ ಬೈ ದ ಕೋಲಿಯೋಪ್ಟೈಲ್ ಆ್ಯಂಡ್ ದ ರಾಡಿಕಲ್ ಇಸ್ ಕವರ್ಡ್ ಬೈ ಕೋಲಿಯೋರೈಸಾ. ಸೊ ಎಂಬ್ರಿಯೋನಿಕ್ ಆಕ್ಸಸ್ನಲ್ಲಿ 2 ಪಾರ್ಟ್ ಯಾವ್ಯಾವು? ಪ್ಲುಮಿಲ್ ಅಂತ ಬರ್ತದೆ, ರಾಡಿಕಲ್. ಪ್ಲುಮಿಲ್ ಈಸ್ going to develop into shoot system, whereas the radical is going to develop into a root system. Then come to the conclusion part. So here when I come for conclusion, after all this fertilization event, we are going to observe the product, that is fruit and seed. Then what is fruit? A fruit is nothing but a fertilized ovary. Fruit ಅಂದ್ರೆ ಏನಪ್ಪಾ ಅಂದ್ರೆ ಓವರಿ ಯಾವಾಗ ಫರ್ಟಿಲೈಸ್ ಆಗ್ತದೆ ಅದು ಫ್ರೂಟ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗ್ತದೆ. ಸೊ ದ್ಯಾಟ್ ಫ್ರೂಟ್ ವಾಲ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಾಲ್ ಇಟ್ ಆಸ್ ಪೆರಿಕಾರ್ಪ್. ಇದಕ್ಕೆ ಏನಂತ ಕರೀತಾರೆ, ಪೆರಿಕಾರ್ಪ್, ವಿಚ್ ಇಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ 3 ಲೇಯರ್, ದಟ್ ಈಸ್ ಔಟರ್ ಎಪಿಕಾರ್ಪ್, middle mesocarp and inner endocarp. So here when I say if fruit is developed without fertilization of ovary, we call it as parthenocarpic. And here there are 2 different types of fruit, one is true and second one false. So what do you mean by true fruit? A fruit which is developed other than ovary. ಯಾವೆಲ್ಲ by the ovary, generally we are going to call it as true. ಯಾವೆಲ್ಲ fruit ovary ಇಂದ ಡೆವಲಪ್ ಆಗ್ತವೆ ಅವಕ್ಕೆಲ್ಲ ಏನಂತ ಕರೀತಾರೆ, true fruit. Those fruit which are developed other than ovary, it may be thalamus, it may be receptacle, that is we are going to call it as a false fruit. For example, your apple, for example strawberry, for example the cashew nut, All these are the example for the false fruit. Then after this, when I come for seed, what is seed? It is a fertilized ovule. Then what is the advantage of seed? Seed having different advantage. Number one, helps in the dispersion. Okay, the transportation of the plant from one area to another area in the form of seed. Then second one, maintaining, or you can say the transfer of character from parent to the offspring, it is carried by the seed. So seed having the future generation in the form of embryo, and it has a nutrition which is going to supply for the developing embryo. So it helps in the dispersion mechanism. So all these are advantages of having a seed. Then in the seed also, so we are going to call it as an important phenomenon called as seed dormancy. So what do you mean by seed dormancy? Seed dormancy, nothing but it is a resting period, or you can say physiologically inactive period of a seed which do not undergo any germination process. Why? Because the embryo is still under the developmental process, or you can say the embryo is still not at all matured. So for that, what happens here? The embryo still in a immature condition, the seed unable to germinate, hence that period we are going to call it as a resting period or a dormancy period. Okay, then so here there is another important phenomenon we are going to observe, apomixis. So remember the tricky point here. When I say apomixis, it is a formation of seed. Okay, it is a formation of seed without fertilization. Okay, seed ಬರ್ತವೆ ಯಾವುದೇ ರೀತಿ fertilization ಆಗಲ್ಲ. ಸೊ ಎಲ್ಲಿಂದ ಬರ್ತವೆ seed ಅಂತಂದ್ರೆ it is from a tissue which is going to encircle the embryo sac, that is a nucellus. A nucellus tissue enter. Okay, the nucellus ಇಂದ ಏನಾಗ್ತವೆ diploid embryo formation ಆಗ್ತವೆ. There is a formation of a seed without fertilization. We are going to call it as what? The apomixis. So here the important point ಏನು ನೆನ್ಪಿಡ್ಬೇಕಪ್ಪ ಅಂದರೆ ಇಟ್ ಇಸ್ ಅ ಸಾರ್ಟ್ ಆಫ್ asexual reproduction. ಅಲ್ಲಿ ಫ್ಯೂಷನ್ ಆಫ್ ಗ್ಯಾಮೆಟ್ಸ್ ಇಲ್ಲ, ವಿತೌಟ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಅದೇ, ಹೆನ್ಸ್ ಇಟ್ ಇಸ್ ಸಾರ್ಟ್ ಆಫ್ asexual reproduction which mimics a sexual reproduction. ಯಾಕೆ ಹೇಳ್ರಿ, end of sexual reproduction flower result in fruits and seed. Similarly, end of apomixis also result in seed, but look at the process, it is completely asexual method. Okay, that is apomixis. Then in case of citrus or in case of mango, what happens here? We know that in a single seed only we can observe one embryo, but here in some plants like citrus and mango we can observe more than one embryo. ಅಲ್ಲಿ ಏನಾಗ್ತದೆ, ಒಂದಕ್ಕಿಂತ ಹೆಚ್ಚು ಎಂಬ್ರಿಯೋ ಇರ್ತವೆ. that condition we are going to call it as polyembryonic condition ಅಂತೇಳಿ ಕರೀತಾರೆ. ಅಗೇನ್ ಎಲ್ಲಿಂದ ಬರ್ತದೆ, ನ್ಯೂಸಲಸ್ ಇಂದ ಏನಾಗ್ತವೆ ಎಂಬ್ರಿಯೋಗಳು ಎಂಬ್ರಿಯೋ ಸಾಕನ್ನ ಎಂಟರ್ ಆಗ್ತವೆ, ದೇ ಫಾರ್ಮ್ ಅ ಮಲ್ಟಿಪಲ್ ಎಂಬ್ರಿಯೋಸ್ ದೇರ್, ಹೆನ್ಸ್ ಇಟ್ ಕಾಲ್ಡ್ ಆಸ್ polyembryonic. ದೆನ್ ಇನ್ನೊಂದು ಲಾಸ್ಟ್ ಟರ್ಮ್ ಏನದು ಬರ್ತದಪ್ಪ ಅಂದರೆ ಪೆರಿಸ್ಪರ್ಮ್ ಅಂತ ಬರ್ತದೆ. when I say perisperm, that is called as persistent nucellus ಅಂತ ಕರೀತಾರೆ. in some plant, okay, in some plant we are going to observe the nucellus present in the seed in the form of a persistent nature. So that is called as what? The perisperm. So these are the important points or a synopsis you have to remember when you go for solving a question. So students, from upcoming videos regarding the same chapter we can expect some important questions to be discussed in an upcoming video. I request you, dear students, please kindly subscribe and share the videos among your friends. Okay, thank you so much.